Okay, law of independent assortment. So this is when we start to come to the point where we're going to learn about dihybrid crosses. So bear with me. So law of independent assortment. Again, this goes back to the whole meiosis incident of crossing over. And what it's saying is that when that crossover happens in meiosis, that the genes are independent of each other. They don't influence each other. So when those genes cross over, they don't interact and influence each other. They are independent. So remember, genes act independently. So then genes act grown, like they know everything, and they are crossing over without influencing each other. And because of that, we can do a dihybrid cross. And there's a picture of one on page 106. So it looks super scary, but the main thing you need to get with dihybrid crosses, you need to memorize this ratio, the 9-3-3-1. So dihybrid cross, remember, this ratio, 9-3-3-1. Okay, so let's try to read the one they've already done to save some time. So they crossed, so this is a cross between, let's see, so they took this guy and crossed him with this guy. So all homozygous dominant to all homozygous recessive. And those traits could have been anything. So let's make it more readable. So let's say capital Y is yellow. Lowercase y is green. Capital R is tall. Lowercase r is short. So you can use them to represent anything, right? And also, like with the dihybrid cross, note that we're talking about the same plant. So I can have a yellow plant that's tall, a yellow plant that's short, a green plant that's tall, or a green plant that's short. So we're looking at two traits of the same plant. That's where the di comes from in the dihybrid cross. So I cross this. So this is homozygous dominant and homozygous dominant. So this dominant yellow, dominantly tall with this recessive green, recessive short gene, and I get this heterozygous outcome. And when you take the heterozygous outcome and you put it on the square, so we doubled up, so we have 16 boxes now instead of 4. You have to do one for each. So you have a dominant, dominant, recessive, dominant, recessive, dominant, recessive, recessive, dominant, dominant, recessive, dominant, recessive, dominant, recessive, recessive. And then you cross them. You bring that into here, and that into here, and that into here, that into here, but you have to put them beside the correct letter. And you get these outcomes of 9, 3, 3, 1. Now, mind you, that is 9, sorry, it's hard to write, out of 16, 3 out of 16, 3 out of 16, 1 out of 16. Okay? And we read it the same way for the genotype and the phenotype. So if I wanted to read this outcome, let's see if we can get it here. So this guy is going to be yellow and tall, phenotype, and then you write that down. This guy is going to be yellow and tall. So again, 2 yellow talls. So you do for each. So you go through how many yellows, how many greens, how many talls, write it down, how many shorts, and you write it down. You count them. And you're going to get this ratio every time. 9, 3, 3, 1. And if you write it as percentage, remember it's 9 out of 16, 3 out of 16, 3 out of 16, 1 out of 16. The main thing is with dihybrid crosses, don't get too wrapped up. Just make sure you know that important ratio and what it means. So, and know that you're looking at two traits. So this represents a trait, this represents a trait, this represents a trait, this represents. So yellow tall, yellow tall, yellow tall, yellow tall, yellow tall, green, tall, yellow, tall, green, tall, yellow, tall, yellow, tall, yellow, short, yellow, short, yellow, tall, green, tall, yellow, short, green, short. So you don't have a whole lot of these, right? You only have one of those. But you heard some repetition in there, so see if you can count them out yourself. Give that a try and see if you can come up with that ratio. That'd be a good practice. All right. And a note, independent assortment is related to the random assortment of your chromosomes and meiosis. So when how they do that random is completely random, and that allows for that variation. That's the same concept. It's not different, it's just under a different discipline, genetics. Okay, so laws of inheritance. So this is just talking about incomplete dominance, which is pretty straightforward. Incomplete dominance means one is not completely dominant to the other. So incomplete dominance is like that original thing we talked about of like putting the traits in there and it makes a smoothie. So incomplete dominance would be like if you have red and white and you put them together. Well, red is not going to dominate to the white and white is not going to dominate to the red. They're going to mix together and give you pink. So incomplete dominance would be if you have black and white. The black will not be dominant to the white. You mix them together, you're going to get gray. So that's what it means by incomplete dominance. And so I would suggest kind of just reading through that and looking at how it's abbreviated. It's a little different. Because one is not dominant to the other, we cannot write it as so how we did before. We kind of have to use these different. So for red flowers, we would put the color that it is. And for white flowers, we put the color up there that it is. So we have white, white, red, red. And when we cross those, we'll get our red, white. But this red, white, because neither one was dominant, means pink. So that equals pink. So let's write that out. So we got CW, CW, which is white. And this is codominant. Oh, Lord. Incomplete dominance. And this is red. And because neither one is dominant to the other, when I cross them, I get this, which is pink. Okay? Codominance. Codominance means both traits are expressed. So the most common example you'll see of this is in your blood types. On the outside, so of your red blood cells, this is a red blood cell. On the outside of your red blood cells, you have these proteinaceous antigens, which determine your blood type. So that's how your blood type is determined, by the type of antigens you have. You can have A, B, or O means you have none. Or you can have A and B. So codominance is this A and B. It's because you can have A and B. So if these mean A and those mean B, this would be what it's like with AB. So you would be AB blood type. They're codominant because they're both being expressed. And we have to also write that a little bit differently, but it shows you right here. So they did, this would be a person with AB. This will be a person with AB. And they just crossed it, AA, AB, AB, and BB, and it shows you that you still get that 1-to-1 ratio. So if you're talking about codominance, you need to memorize that 1-to-1 ratio for codominance, because that's what you're always going to get. So we're taking 2 traits expressed on this one, 2 traits expressed on this one. We're crossing it, A and A, B and A, B, A, B, B. So you got 1 AA. You got 2 ABs and you got one B. So these are the chances. So if you and your husband are both AB blood types, this is your chances of having a child with AA, AB, or BB. This is not factoring in O. There's a way to do that, but you ain't got to do it in this class. So, okay. Wild type, and I'm on page 109. Wild type is just the most common way that gene is found in the wild. Wild doesn't mean wild as in like the crazy form. Wild is actually the most common form. Wild type is how it's found in the wild. And here they factored in the O. So they took this little I to represent the O. So the O is your chances of having no protein on the outside. That's why O is the universal donor, because people's blood types don't fight against those protein antigens because it doesn't have any. Because if you put the wrong blood type in a person, you have, so like if you're type A, you're going to have anti-B antibodies. So if you put B in your body, it's going to fight, your body's going to clump it, and it's going to possibly kill you. So that's why blood typing is really important if you're going in the medical field. But read up some more on that. There's a lot to it. It's interesting. Learn about it in anatomy more. But anyway, so they just crossed an AB person with this chance of having O, and if you see, it comes out to a lesser chance of having O. But like a person can have, like if you have this chance of O factored in, that means your child could be A or AO versus A. So there's a lot to that. I wouldn't get too caught up in it. If you would like, there are YouTube videos that work with blood typing. Because both of those ABs, while they're co-dominant to each other, right? Because neither one dominates, they are both dominant to this O. So A and B are equally dominant, but they are both dominant to O. So this person would have O. This person would have B. So O. Because all they have is that recessive form. They have a B and a recessive, but B's dominant. They have the recessive and an A, but A's dominant. They have the recessive and a B, but B's dominant. They have two B's, so B. They have an A and B, well, they're equally dominant, so both. Just an A, so A. They have an A and B, which are equally co-dominant, so both. And an A and a recessive O, so A is dominant. So if you put A or B up against O, it's showing you that they are dominant to it. So that's the main thing. Sex-linked traits, I encourage you to read about it on your own. It's pretty straightforward. Make sure you know about linkage and hemizygous and X-linked, those bolded words. Just make sure you have a pretty good understanding. If you have questions, let me know. Epistasis, this is just talking about gene expression. So, polygenetic inheritance. Poly means many. So skin color, human height are good examples of that. So that's when many genes are coming into play. That's where it's in polygenetic. Many genes are affecting the inheritance of the trait. So if you see poly, it's talking about many genes. Epistasis is when the genes are antagonistic to each other, so one gene is canceling out the other. So if this certain gene is being expressed, then another gene will not be expressed, and it uses this as mice. So if a certain color gene in the mice is expressed, it cancels out the expression of another, which leads to these outcomes. So read about that. It's pretty straightforward. Let me know if you have questions.